All right, let me just start the timer. Okay, awesome. Well, hello everyone. My name is Olivia Bauer-Nilsen, and I am a master's student at the University of Cincinnati in the UTS lab. And today I will be talking to you about chemical communication in Schizocosa ocreata. I'm sure that many of you are familiar with the brush-legged wolf spider, but for those of you who aren't, Schizocosa ocreata is a well-studied wolf spider species due to their multimodal communication and their complex mating system. Males possess tufts of black bristles on their forelegs that serve as secondary sexual traits that females find attractive. Courtship consists of both visual and vibratory components, as seen in this video. First, the male detects the female cues by chemo-exploring, and then subsequently begins his courtship display. So you can see he's waving his arms and tapping both his forelegs and his body, creating a vibratory and visual courtship signal. Studies have shown that tufts are condition indicating traits, and they reflect juvenile foraging success or nutrition and immune strength. And we've shown that females prefer tufts that are larger and more symmetrical. In this presentation, I will focus on the eco-immunology of Schizocosa ocreata in the context of chemical communication and sexual selection. So a previous PhD student named Rachel Gilbert found that infection by Pseudomonas aeruginosa, a common arthropod pathogen found in all environments, including the leaf litter where ocreata are found, will affect the behavior and development in males. Males who were infected as juveniles developed smaller and more asymmetrical tufts as adults. And subsequently, juvenile infected males also had reduced mating success. As adults, though, male tuft size is fixed, and males infected as adults increased their courtship vigor in mating contexts, which could be a potential compensation strategy making up for their compromised health. The talk right after this one will get more into this compensation strategy seen in males of this species. So look out for that one. Females, on the other hand, show reduced locomotor performance when infected as juveniles or adults, demonstrating the physiological and energetic cost of infection. In this locomotor experiment, males showed no significant reduction in locomotion performance when infected. And this shows that trade-offs with immune function are different between the sexes. When males begin searching for mates, they will pick up on the chemical cues associated with a female's dragline silk. Many studies in Schizocosa species, as well as other members of the family Lycosidae, have found that males can discriminate between females of different status using these chemical cues alone. Information contained in the chemotactile cues can inform male behavior. Whether a female has mated previously, her age, and her feeding status are some factors that will affect the vigor with which a male courts. And males don't produce much silk as adults, but they still leave cuticular hydrocarbons behind as a chemical signature. However, it's still unclear how and if females respond to these chemical cues of males. But Rachel Gilbert aimed to test this by presenting females with a two-choice test of chemical cues from infected and control males, associated with video playback of courting male stimuli as shown in this video and diagram. With all factors held constant other than chemical stimulus, Rachel found that females performed more receptivity to the chemotactile stimuli of the control male. This suggests that females do discriminate between male chemical cues in a mating context, at least with some courtship stimuli present. But what I want to know is, what about with chemical cues alone? Which leads me to my main research question, which is, can spiders of either sex detect pathogenic infection or immune challenges through chemical cues alone. Here's a video of a male chemo exploring on female cues. We have evidence that males can discriminate certain aspects of female status from chemical cues alone. And we know that females show more receptivity to control male cues when presented alongside visual courting male stimuli. But we still have some gaps here, and that's where I come in. I want to fill those in. And to do this, I infected both males and females and collected their cues to present to the opposite sex in behavioral trials. Okay. First, I wanted to dig deeper into what Rachel found with female preference. So to start, I infected males as juveniles in order to induce an immune challenge by withholding water for 24 hours and presenting them with a 1 microliter droplet of either sterile water containing 600 colony-forming units of the bacterial pathogen Pseudomonas aeruginosa, or for the controls, just a 1 microliter droplet of sterile water. When these males became adults, their chemical cues were collected over 24 hours on filter paper. Then to test, the spiked filter paper was placed into a 2-chambered arena, as shown in this diagram and photograph. One side of the arena, which was randomly chosen per trial, contained the previously male spiked filter paper, and the other, a blank filter paper. Females were introduced into the blank side of the arena and allowed to explore for 15 minutes, while trials were recorded and later scored for visits and time spent. Results found were overall non-significant. So females visited both infected and control male spiked filter paper with close to equal frequency. And although females spent more time on the control stimulus in total, they opposingly spent more time per visit to the infected stimulus. However, both measures were insignificant. So overall, no significant trends in visit, time spent, or visit duration were observed. So females are not discriminating when given male chemical cues alone. And I just want to hit this home really hard, because what we see is that females can use chemical cues to inform their behavior and subsequent mating decisions when presented in a mating context, aka with chemical cues associated with a courtship stimulus. But chemical cues of males singly are not significant or sufficient to elicit discriminatory behavior in females. Maybe it's just not worth it if they don't know a male is available and nearby. I have some ideas to test this further, which I'll get into later. But now let's switch over to the male side of things. But the main takeaway here is that male chemical cues are not sufficient to influence female behavior on their own, although they do when associated with the visual courtship. But to switch over to our male side of things, we know that males can use chemical signals on their own to inform their courtship effort, but we don't know if males can detect an immune challenge or infection in females and use that information to alter courtship vigor. So similarly to how we infected males, adult females were withheld water for 24 hours and given a droplet of water either containing the pathogen or not for control. 1 hour post-treatment, females were placed in an arena and allowed to lay down dragline silk and chemical cues. For testing, the female spiked filter paper was placed in a 1-chamber arena where a male was introduced and allowed to explore for 10 minutes while being recorded. Later, the videos were scored for courtship behaviors and bouts of courtship. Some of the behaviors are depicted in this diagram, which include body bouncing, tapping, double tapping, and the wave arch. And lo and behold, males have done it again. Males showed greater courtship effort when exposed to control female silk compared to infected female silk. And specifically, males performed more of the costly body bounce behavior with a p-value of 0.0256. This is really exciting to discover and provides yet another aspect of female condition that males can use to inform their courting effort. So looking to the future, there are more gaps to be filled into our knowledge base of chemical communication in Schizocosa ocreata, and Although these signals are all but invisible to us, they provide a robust source of information for males choosing where to invest their energy. Chemical cues are also potentially a great source of information for females deciding whether or not to mate. For example, do females discriminate between infected and control male chemical cues if presented with vibratory courtship stimulus, not visual? In the same vein, what would happen if females were given a multimodal signal with both visual and vibratory components associated with male chemical cues? Would discrimination be even more extreme? And the most pressing question for me is, what would happen if females were given a direct choice between control male spiked paper or infected male spiked paper at the same time? This paired experimental design has been completed this summer, and I'm working on scoring it now, so hopefully I'll have more on this really, really soon. But with that, I would like to begin my Arachknowledgements by thanking my advisor, Dr. George Utz, who has been such an inspiration for me and a support as a researcher and an arachnologist. I'd also like to thank Dr. Brent Stouffer for invaluable input on experimental design and being a great resource for my stats questions. I'd also like to thank previous undergrad Manny Bagarov, who helped me score the endless number of male courtship videos I had. Thanks to all the undergrads who have helped collect and maintain our spider population, and of course to the spiders themselves who sacrificed themselves to be part of the study, as well as, of course, the American Arachnological Society for funding and support, and the Animal Behavior Society for funding. Thank you to the Cincinnati Nature Center as well for allowing us to collect on their land. And to follow suit of some other colleagues, I wanted to acknowledge the original peoples who lived on this land long before us. But thank you all for listening to my talk, and I'll take any questions.
All right, there's a few questions in the chat, so I'll get right to them. So how long are the chemical cues present in the silk before being undetectable by the spider?
So the chemical cues are pretty much there until water. So in the field, this would be after a dew. So overnight, when dewdrops form, the water will basically neutralize the chemical cues, and after water has been applied to that silk, the chemical cues will basically be neutralized or washed off.
Great. So did the males that you had producing the cues know that a female was around? Did he have any indication there was a female present when he was?
No, it was only her chemical cues. Or wait, are you saying when he laid the silk originally?
That's correct. Yeah, that's my understanding.
Yes, no, there was no female around. He was just set in an arena with filter paper and allowed to kind of wander around for 24 hours. And I had them laying down chemical cues for a significantly longer period of time because they don't lay silk as dragline silk as much as a female would.
All right. And how did you isolate the chemical cues from the silk? Could it actually be that physical cues of the silk are the important cue instead of chemical, i.e. increased number of anchor discs?
Yes, so it's possible. The chemical cues and the silk were not separated. So in my study, I basically was considering silk and chemical cues as one stimulus, because in previous studies we have found that the chemicals and the silk they will both elicit the same reaction. So Rachel Gilbert, actually, when she did her study with the female's reaction on male chemical cues, that was a big question for her, because since males don't lay down a lot of silk, we were wondering, like, are the cues even going to be there? Because if they don't lay down silk, where are the cues? But she actually found that if you just rub a male, his body surface, on a piece of filter paper, or dip him in ethanol for a certain amount of time, that those chemical cues on the cuticle come off, and then a female will respond equivalently to those chemical cues without silk.
Cool. All right, there's a bunch more questions, but we won't have time for them. So I would suggest that you contact Olivia directly, or,
We actually have time for another question or two.
Oh, we do.
Yeah, the other track's a little behind.
Okay. I guess we're still trying to match. Okay, one more question, sorry. And is there a way to confirm that the spiders were infected by the bacteria? Could possibly they not be infected?
Yes, so they could, I suppose it's possible, but this is also another kind of some of the heavy legwork that Rachel did in her PhD, was that she was the one who discovered that 600 colony forming units is enough to elicit a reaction and an immune response, but not kill the spider. And she also was able to find that when she infected them, she did an assay of their hemolymph over, I think, 7 or 9 hour period. And she was able to find that within the hemolymph, between like 1 and 3 hours, the bacteria concentration in the hemolymph was the highest. And then by 5 hours, it was significantly reduced. So the spiders are actively fighting this infection during this 7 hour period. So after 7 hours, it's pretty much eliminated from the hemolymph. Basically, it's just like at basal levels. But so we are sure that there's some physiological immune response that is being activated here. And she also looked at their encapsulation, but not, I don't think, during an active infection.
All right. So